a safer, natural alternative to the weight loss peptides like Ozempic and semaglutide, retatrutide, and tirzepatide? Although these GLP-1 weight loss drugs are taking the world by storm because they work, is it possible that they come with downsides? And what if there's an alternative that few people are discussing? Well, scientists, like the one we have on today's podcast, say that there are. Join me and Dr. Edward Walker from Calocurb as we discuss a substance called Amarasate, which is a powerful plant extract that naturally stimulates GLP-1, but significantly. It helps manage cravings and balances your metabolism without the risk of pharmaceuticals. But is it too good to be true? Stick around as we break down the science, the benefits, how you can start using it, and whether or not this more biocompatible substance really works. In this episode, we discuss the impact and side effects of things that increase GLP-1, what scientists look for when they're evaluating compounds that naturally increase GLP-1, because there's a difference between a statistically significant increase and a clinically or outcome significant increase. We explore the benefits and side effects of Amarasate and some different synergistic stacks you can use to increase GLP-1 levels and blunt cravings, increase satiety, and do everything else, potentially lose weight, and do everything else that people are turning to the GLP-1 drugs for. Dr. Ed Walker is a scientist and lecturer specializing in plant-based nutraceuticals for health and wellness. He earned his PhD from the University of Auckland, researching the antioxidant effects of berry fruits, and now works at the New Zealand Institute for Plant and Food Research, focusing on clinical validation of nutraceutical products. Most relevantly, his 13-year research on plant-based appetite suppressants led to the development of Amarasate, a New Zealand hops-based suppressant with a mode of action similar to GLP-1 anti-obesity drugs. All right, to be clear, I think that the GLP-1 drugs that are so popular have their time and place, and they really can change lives when used by the right population. But for healthy populations, I think semaglutide and the newer analogs like tirzepatide and retatrutide are overused, and the healthy folks have better options. If you want to explore the potential downsides that are less discussed about the powerful GLP-1 peptides, I will put a link to an article I wrote in the description of this episode. I also wrote an article listing over 45 different natural alternatives you can use instead of those peptides, one of which is Calocurb. I actually have it right here in front of me, but I actually didn't notice much, if any, effect from one capsule, yet when I increased my dose, all of a sudden that changed. So we get into the nitty-gritty of dosing and the bioindividuality of it later in the show. After we recorded, I also just came across this study. Haven't even read the whole thing yet, but it is titled Effect of Glucagon-like Peptide 1, GLP-1, Receptor Agonists and Coagonists on Body Composition, Systematic Review and Network Meta-Analysis. Although I haven't read it yet, and I'm looking forward to digging into it, the conclusions were that tirzepatide and semaglutide, two of the most popular GLP-1 drugs, have a stronger weight loss effect, but also cause a significantly increased loss of lean body mass. Now, this is in comparison to the other GLP-1 drugs, and well above what happens with standard weight loss. So yes, these very powerful substances have their time and place, but before going to the big guns, they work incredibly different to the way natural GLP-1 works in the body. Consider the more natural approach. If you want to learn more about this, you can find everything we discuss in the show notes for this episode, which will be at mindbodypeak.com/193. And if you want more details on weight loss, the GLP-1s, peptides, or any other topic, check out the free Mind Body Peak Performance AI chat. Go to mindbodypeak.com/AI and get all of your questions answered. It's like having me and my guests available to you at all times. All right, ladies and gentlemen, sit back, relax, and enjoy this episode with Ed. Ed, welcome to the podcast.
Hi, thanks for having me.
This one has been a long time in the making. We're going to be discussing your star ingredient that you've been researching for nearly, I think it's a decade and a half at this point. Tell me how you got involved with the substance that is known as Amarasate.
Right, yeah. So it all started back in 2009, and what we had was the New Zealand government put out basically a call for research programs to develop a plant-based treatment for obesity and appetite management. And so I was employed basically by a government institute to do that research. And what basically happened was we came up with a series of ideas about targeting the gastrointestinal tract to suppress appetite. We put in a grant. It was a 6-year, $20 million government research grant. And we got the funding, and it sort of kicked off 2010 and went from there. So I started there. Immediately before that, I just finished a PhD in molecular medicine looking at the health effects of berry fruit. So I sort of had, I guess, a background in plants and health, and then straight into this weight loss project.
Awesome. And I think you were one of the early researchers of a now famous class of drugs called the GLP-1 agonists, the semaglutides and Wegovy's and all that stuff of the world. What made you fascinated about this class when you started researching natural substances that may activate the same or similar pathways?
Yeah, absolutely. So we had an idea, right, that we could suppress the appetite using hormones from the gastrointestinal tract. So every time you have a meal, right, your body will release a series of gut hormones that tell you you're full and you've eaten, and they're primarily CCK, PYY, GLP-1. Right? And they all have different sort of benefits and downsides to them. And CCK, for example, is a really amazing appetite suppressant. It kicks off really early, suppresses your appetite hugely, but it tends to have a rebound effect, so it's not very long lasting. So that's not so much a really good drug target in isolation. When they did work on GLP-1, so GLP-1 was discovered sort of 1980s. What they found in mid-90s, they realized it was an appetite suppressant. What they found was when they did what they call infusion studies, so basically they'll get people to walk around with a bag of GLP-1 and they would infuse it into their blood over time. They found that they could hugely suppress appetite, and it was long-term. It would reduce their glucose levels as well, and it could even lead to weight loss. So there was a real, I guess, understanding from around the mid-90s that GLP-1 was a really good target for weight management and weight loss. Now, the pharmaceutical side of things, what they said basically GLP-1's not very stable. Right? So in the body, what happens is you have a meal, GLP-1 gets released, it gets released. As soon as your meal's gone, goes back down again, and that tells your body to eat again. So the pharmaceutical industry said, let's stabilize it, right? So let's modify the GLP-1 molecule and make it stable, make it last for a week rather than, a few hours in the body. Now, that's great for a drug, but what it does is it takes a natural occurring hormone and changes it into something which is not following temporal profiles. It's not going up and down with your body cycles. It's not resetting at night. It's just a constant high level, 24 hours a day, 7 days a week. And, that's not physiological. It causes your body to downregulate its natural hormones, just like, say, hormone replacement or steroid abuse would do. So there's a series of downsides. So we really said, well, pharmaceuticals doing that, that's cool. Why don't we look for a way just to get your body to produce more GLP-1 and to release more GLP-1 so that you can use your natural system, you can boost up your natural system rather than trying to just completely put something artificial version into it. And so that's sort of, I guess, where we came in it from. And we sort of thought about a few different ways of trying to trigger GLP-1. And indeed these other appetite suppressing hormones. And we really came up with the idea that gastrointestinal bitterness was a really, I guess, potent way of triggering GLP-1 release. And that was sort of the kickoff of the project for me.
I want to pause you there and go more into these substances that pharmaceutical companies have developed to potently increase GLP-1. Also, let's back up a little bit. You mentioned the purpose of GLP-1. What are these peptides, the ones that I mentioned previously? Why have they become so popular?
Do you mean the injectable peptides?
Yeah, the injectable peptides, and they're now, what was off-label use is now probably one of their main uses.
So what GLP-1 does in the body, right, is it is a signal to your body that you have consumed food and that you're likely to have glucose coming in and that you're likely to not need to eat for a while. So it releases from your gut, right? It goes to your pancreas. It will stimulate insulin secretion to lower your glucose levels. It'll go directly to your brain and it will suppress the appetite centers. It'll also hit what's called the vagus nerve, which is a nerve that runs from your gut to your brain, and directly trigger that to suppress appetite. So what it is it's a small peptide molecule, so small protein molecule. It's around sort of 30 or so peptides long. I mean, 3 to 37. So there's different forms of it. But what it has on it is it has a cleavage site for an enzyme called DPP4, right? And what DPP4 does is it cuts that GLP-1 into a non-active form really quickly. So the half-life in the body is 2 minutes. And what that means is that if you're not actively releasing it,
natural GLP-1,
this is the natural one, natural GLP-1, yeah. So it lasts 2 minutes. So if you're not producing it, it's gone. And that's really important for your body because it means that you can, make yourself feel full after a meal, have a proper glucose response after a meal, but not have a constant stimulation of the system. Right, so what the pharmaceutical industry did was they did a couple of different things. Firstly, they said, let's stabilize it by binding it to albumin. So that's like, sort of some proteins in your blood. And if you bind it to albumin, it makes it harder for this enzyme to access it and cut it. Right? And so that was liraglutide. So that was the first version of it, of the, I guess, the commonly known GLP-1 agonists, or the pharmaceutical versions. And so that increased the stability to half-life to about a day, right? So about 24 hours around that. Then they may come up with semaglutides. And what semaglutide is they actually actively modified the enzyme cleavage site. So they didn't just bind it to the albumin to make it harder for the enzyme to access it. They modified the site so the enzyme can't really cut it very well. And that extended the half-life to around a week. So you've gone from 2 minutes naturally to 1 day to 1 week for the sort of more recent semaglutide type drugs of Wegovy. Yeah.
And that right there is a huge difference. It might sound minor. I heard a doctor call the GLP-1 drugs like that one you just mentioned bioidentical to natural GLP-1, except that it lasts a bit longer. And when something lasts 6,000% longer, it's not bioidentical. It has totally different effects on the biology and all the different timings and rhythms that are naturally happening throughout every hour, day, even a week.
Yeah, absolutely. I mean, you could say if someone comes along and says, well, I'm an anabolic steroid user and it's exactly the same. Like I can take Tren and it's fine because it's bioidentical. I mean, everyone knows that's ridiculous. And so that comparison is clearly not right. One, it's, the binding efficiency is much higher, so it's stronger for starters, and the temporal profile is completely different. Yeah, so it's absolutely very, very different indeed. And I think that a lot of this was probably driven by marketing requirements. Because when you think about you've got an injectable drug, you're asking yourself, how frequently can we ask someone to inject a drug? Right? So they could have, and there were versions of these pharmaceuticals or like trial versions that were less stable, which might have had a stability of a number of hours or but really when you look at it, injectables once a week was sort of, I guess, my thought is this was the key. Someone will inject once a week, but they don't really want to do a lot more than that.
What are some of the other effects this has on the body? You mentioned it also causes changes in the brain, which is why we're also seeing it being used more for addictions. That's one of the other hot use cases for it, because I think it triggers what are called the POMC neurons.
Yep. Yeah.
Break down other effects. So you mentioned that and the vagus nerve. And ironically, I've talked to a number of different users who, when they measure their vagus nerve via something called heart rate variability, they see dramatic reductions. And that's not across the board, but for a lot of people, and I've seen research on this too, that they have impaired resting heart rate and other proxies of recovery.
Yeah. So, I mean, the addiction is probably twofold, the way it's affecting that. And there's the POMC neurons, and there's also likely to be changes in some of the neurotransmitter levels that are linked to the addictive behavior, things like dopamine. And so, certainly, there's been indications of GLP-1 being a target molecule for addiction, but also for things like behavioral disorders. So, a long time ago, there were indications that GLP-1 treatments via infusion, so this is the normal GLP-1, not the stabilized version, would cause a acute increase in anxiety and then a chronic decrease in anxiety, right? So there was sort of like this, I guess a U-shaped response. And so that long term there might be anti-anxiety effects, and long term there's likely to be anti-addiction effects as well through those addiction pathways in the brain. And I think that's one of the really strong, one of the reasons why it works so well, why these injectables anyway work so well, is because they're treating not just biological hunger triggers, but they're also treating food addiction triggers. Yeah, and that drives a lot of people, because you can, if someone's eating when they're not hungry, then it's really challenging to change their eating behavior. But if you manage to suppress that, then that's really strong. And for the vagus nerve, absolutely. So changes in heart rate variation is something that you can measure. We're actually running a study right now that measures changes in heart rate variability. So we're looking at giving in one part the Amarasate extract, and we're looking at changes in vagus nerve activation via a measure of heart rate variability, and we're also measuring neurotransmitter levels, peripheral measure of neurotransmitters. So looking if we can do the same kind of change with a natural product that you're seeing with the pharmaceuticals. So so it's really exciting, and so GLP-1's amazing. Yeah, it really is.
There's one other mechanism I want to dig into with you, because this is the one that when I saw, it gave me pause. I wrote an article, I want to say 3 years ago, when I first started reading the research on these things, and the synthetic GLP-1s that are modified to last way longer, they have an impact on insulin. And insulin in the longevity space is like public enemy number one. Yeah, we also know this is stimulating insulin, and it's doing so for a week, 160 hours, I believe it is. Tell me about what this is doing. I actually have a paper up in front of me. It's called Regulation of Adipocyte Formation by GLP-1/GLP-1R Signaling. What happens when you signal GLP-1 and insulin like that for 160 hours?
Yeah, so it's an interesting one. So there's two aspects to GLP-1 and insulin, and the main one that people will talk about is that it's an insulin stimulator. So you inject GLP-1 and you stimulate insulin release, and that's absolutely true, and it's really quite potent. There are some studies that also show that GLP-1 is an insulin potentiator, and what I mean by that is that it will increase the activity of the effectiveness of the insulin release per unit of insulin. So if you have 10 units of insulin, or arbitrary number, it's slightly better at removing blood glucose than it would otherwise be. And they do that based on some clamp experiments in rats. So they basically isolate part of the rat, and they'll put in a fixed volume of glucose and insulin, then add some GLP-1 and see how quickly it's removed. So there's that aspect to it. But you're absolutely right. There are questions around constant stimulation. And I think these likely feed into some of the reports of more rare side effects you may have with the GLP-1 injectable. So if you think about your pancreas is used to getting a GLP-1 signal every time you have a meal for a few hours to release insulin. Now, if you take an injectable, it is getting that signal 24 hours a day, 7 days a week, saying release insulin, release insulin, release insulin, release insulin, right? Now, you do get reports of pancreatitis, right, with use of these, and they're very rare, but they are, I guess, I can't comment on how related they are from a legal perspective, but from a scientific perspective, given that you're stimulating the pancreas a lot, it's possibly not unexpected that this might happen. Right? You also have the same effect with delayed gastric emptying. So usually GLP-1 will go to your gut or your stomach and tell it not to empty for a while. Right? So usually that happens for a few hours. Now you're getting it 7 days a week, 24 hours a day. And we do hear reports of gastroparesis type symptoms really occurring sometimes from injectables. And again, possibly not surprising that this could occur if you're getting a constant signal. So there's absolutely those. As far as things like adipocyte differentiation goes, absolutely. Yeah, so there's certainly a potential for increasing adipocyte differentiation. You're getting a lot of signals to your fat cells to basically say, hey, look, we've got nutrients to uptake. We need more of you to be there. So that's not a surprising side effect. And I guess the concern comes in there for, and this is a concern I personally have, is that if people are using injectables almost like a yo-yo diet, right? So if they're taking the GLP-1 injectable, they're losing the weight, right? And then they're going off it and they're gaining the weight back. Have they just from using the injectable sensitized their fat cells to be, really responsive to uptaking nutrients? And if so, are they just going to make their yo-yo dieting worse over time? So there's absolutely, and you're absolutely right. There are concerns around that, and we'll know in the long term, I guess, what's happening.
So to summarize what you're saying, is it possible that by using these, we're actually making the fat cells in a way more aggressive towards uptaking things, towards growing more rapidly?
Well, I mean, it's absolutely possible. Certainly, when you look at a cell and you want to turn. So if you go into what they call cell culture, so you grow some pre-adipocytes in a lab, and you want them to turn into a full adipocyte or a full fat cell, you will use insulin to do that, right? So if you're stimulating your insulin levels, then that's entirely a possibility. Yeah.
Yeah, and there's a paper on this too called GLP-1 signaling in regulation of adipocyte fat cell differentiation and lipogenesis, which that's a 2017 paper. So it's been around for a little while, and I'm sure there's more that I haven't even seen yet.
Yeah. Well, there's, it's an interesting comment you make there, because it's, when you have a big pharmaceutical drug, there's usually, one paper that'll show something, but then there's not usually a lot of follow-ups because it's quite hard to get funding, and the drug companies are not going to pay for it. So there's also, so there's that adipocyte differentiation issue, but there's another one of downregulation of your natural hormones, and just like happens when you take say exogenous hormones of some form, and you'd think there'd be a whole lot of papers on that, but there's really just one, and it's like, and it came out about 2020, and it says, oh yeah, if you take liraglutide, your endogenous levels are suppressed within 3 days completely, pretty much. And you're like, oh, well, that's not surprising. But there's no long-term looking at how fast they rebound, whether you've got any changes in your stem cells in your gut. These are all pieces of work I would do if I was developing a product. I'd be saying, okay, we've got no GLP-1 in the gut. Let's just make sure that we're not changing our gut stem cells so that they can't produce GLP-1 in the future, and that when we take someone off, it rebounds, but that data doesn't exist yet. Yeah.
Yeah, and these things can be life-saving for people in certain circumstances. They can make dropping 100 pounds for someone who's obese feasible and possible. And it's probably worse to walk around with an extra 100, 150 pounds on you than it is to use a peptide, because we know very clearly that carrying around tons of extra body weight, especially if it's a lot of visceral fat, is extremely unhealthy. And that's one of the big risk factors for all-cause mortality. So there's time and case for it, but I think there's also a lot of other alternatives. Can you touch on the different generations? Because I know that there's multiple different generations, and I think retatrutide is the new one that's on the rise, and it's not actually released yet, but it's like a triple antagonist. Can you break down what the different ones are? And
right, yeah. So initially there was the GLP-1 based agonists. So they were based on essentially just GLP-1, and that was sort of liraglutide and then semaglutide, which have been more stabilized. They've got trizepatide, and what that is that's a GLP-1, and it's another gut hormone called GIP bound to it. So it's basically a dual agonist. And what GIP does is kind of pretty similar to what GLP-1 does, to be fair. It also has insulin stimulating effects, and it also has appetite suppressing effects. And I think that when you look at the combination of the two, so if you look at the data on, say, efficacy, the the two agonists, the dual agonist is more efficacious. But I really think that it's more efficacious for a given side effect profile. So what you really see is you see that adding the extra agonist means that you can keep the side effects at a still quite a high level, but at an acceptable level for more weight loss. And if you just gave more of a GLP-1, you'd probably of semaglutide, which is a single agonist, you'd probably be able to generate that same weight loss. You'll just end up with more side effects. The reason for that is probably because when you look at injectable, when they did studies with the endogenous hormones and they injected them, they got a profile of activity. So from like no effect to appetite suppression to GI side effects. And so if you imagine you've got one hormone, the higher you push it, it's you get appetite suppression, you get sort of side effects. If you've got two, you can have less of an effect on both of them. So so I guess it's a bit more of a gentle way to do it. So then there's triagonists coming out now. I'll see if I can remember. I think it's an amylin. So there's a few different ones, but then there's GLP, GIP, and then I believe they're adding amylin to one, and there's another one with I believe glucagon added to it as well. And so glucagon is another GLP-1 related peptide, which is released from the alpha cells of the pancreas. So it's a pancreatic hormone, and again, it has sort of similar effects. So they're really just trying to and some similar effects, but it's almost like glucagon is almost like a balanced insulin as well. So in some ways that they may be adding this to try to counteract the overstimulation of insulin that they may be seeing, or the overstimulation of insulinogenic effects, right? And so we're likely to see an increase in efficacy and a slight decrease in side effects occurring when they introduce those triagonists.
Yeah, when I saw them adding glucagon activity, I thought that was interesting because it reminds me kind of like getting in a car and hitting the gas and the brakes at the same time, because glucagon brings up blood sugar. It does the opposite of insulin. Yeah,
absolutely, yeah. And so that would be what I would think they were trying to do, is trying to acknowledge that there's maybe too much for if their target is weight loss. Right, they're probably getting too much insulinogenic activity, and they're trying to basically pull it back a bit with the addition of the glucagon. Yeah,
that's interesting. Have you seen any research on what other hormones these might affect? I'm wondering now if there's going to be an effect on cortisol or anything else that you wouldn't necessarily want to stimulate, especially not long term.
Goodness, yeah. So I can't think of anything off the top of my head. I mean, it's pretty likely that those will go up. I think that there's not enough research to really show what the long-term effects of these hormones are. What I'd say is that, I mean, I would expect you to have probably a stimulation of cortisol, but I can't say I've seen a paper. I'd also guess be somewhat concerned that you're going to cause, a systemic suppression of some of your gut hormones all the way down, and a down regulation of not just the ones you're targeting. So for example, when they gave, on the one study they did looking at down regulation, they gave the GLP-1 agonist, and they saw complete down regulation of all the basically glucagon related genes in the gut. So there's sort of two pathways. All the glucagon related genes get processed to, one's in the pancreas, one's in the gut. And so they saw GLP-1 go down. They also saw GLP-2 go down, which is, they're not injecting GLP-2, but it's being downregulated anyway. So I would not be surprised if, as we expand the number of agonists that we put in, we're seeing a more systemic downregulation of a wide variety of endogenous hormones. And there's, I guess, a question mark over what the long-term effects are going to be.
I'm with you on that. I want to pivot now to Amarasate, your ingredient, because when I came across this, I was immediately excited. It's a needed substance in the world. I love it because of the more biomimetic half-life, and the dosing curve aligns much more closely with what you could naturally get. It's still a little more than you can get from a meal, and we'll talk about that. But you're not going to be stimulating insulin late at night at 3 a.m, 5 a.m, 6 a.m, when you're supposed to be sleeping. What sets Amarasate apart from the other powerful GLP-1 activators?
Yeah. So, I mean, to go back to the start of the story, we were looking for a GLP-1 booster, and we did this project called Foods for Appetite Control with the New Zealand government. So I'm a New Zealand government employee. I don't work for the company that sells any products. And what we found out was that gastrointestinal bitterness was a good, I guess, target for stimulating GLP-1 release. So we really wanted to boost up GLP-1, and we wanted to find a way to do it really potently. And so we did a pharmaceutical type approach. We screened a whole lot of a whole lot of extracts. We found a hop extract that we named Amarasate. We tested it in cells. We tested it in people, saw GLP-1 release. Now, one of the things, I guess, that's quite important when you look at like nutraceutical GLP-1 activators, what they call, is that there is it's really easy to get a cell in the lab to release GLP-1, right? So if I grow what you call a enterendocrine L cell, which is the human cell which releases GLP-1, what it is, it's like a circle, and it's got these little vesicles in it. And if you put on a trigger, like Amarasate, those vesicles will go and they will release into the growth media around the cell. Now, the other thing you can do is you can sprinkle some dust on that cell, and it will die. They're super fragile. And when it dies, it will release all that GLP-1 into the media. So, you can publish a paper, and if it's not well done, you can show anything releases GLP-1, right? So you have to be very careful with the science. So if you see GLP-1 release in a cell system, in a cell assay system, then that's not really any proof to me that it's going to work in a person. Like, it's preclinical. It's like, that'll be interesting. That, if you've done it correctly, go forward and do a clinical trial. So lots of the GLP-1 activators out there will have that sort of evidence, right? Where they've just done it in a cell, and it may or may not be a real effect, depending on how well they've done the science. What Amarasate has, which is different, is we have a fully double-blind controlled placebo trial, that shows potent GLP-1 release, and not just a small amount, not like, you've given in say a green tea extract and you've shown a 20% increase in GLP-1 levels. What we're seeing is basically a doubling of your response to a meal. So usually when you have a meal, GLP-1 will go up by around threefold, and that's exactly what we show in our study. But when you take Amarasate an hour before that meal, it goes up by about sixfold. Now, that difference is really important. And the reason it's important, because if you look at the amount of GLP-1 you need in your body to have an additional biological effect by itself, it's around 4+ fold over baseline, right? So we're in that bioactive range for GLP-1. We're also in that bioactive range for some of the other gut hormones, such as CCK, which suppresses appetite. And most of the time when you look at a study, it will not have anywhere close to that sort of change. So it'll have this really small stimulation of GLP-1, and it's just not enough to have a meaningful effect in people.
Yeah, I don't think you know this, but I did some research and I did a roundup of all the different substances and foods I found that can stimulate GLP-1. And there's like 45 of them that I came across that had like some level of improvements. But what you're saying right now is really important. It's not that it had a 5% or 10% or even 30% improvements in GLP-1 release, because that's not enough to actually cause a meaningful, like, clinical outcome. What you need is to have a massive 4x spike over baseline to actually change behaviors in a way that, like, people are seeking when they're using GLP-1 products.
Yeah, absolutely. And that sort of, it was really interesting when I guess this is the benefit of having lots of history of research of GLP-1, is that you were able to figure out the exact amount you need, because they did the studies where they got a bag of GLP-1 and they infused it into people and they measured and they said, is it having an effect, yes or no? And if they weren't going up to fourfold, then they didn't do it. Yeah, so it's and that work was actually done by Professor Sally Poppet, or the latest review article that sort of summarized it was. And she's someone that I've worked with in the past, and she was actually involved in the development of Amarasate as well. Yeah.
Amazing. Tell me more about the actual use of Amarasate, because I've actually used one of the products called Calocurb that's an implementation of Amarasate that contains Amarasate in it. I didn't notice any major changes when I consumed one capsule about, I think it was 60 to 90 minutes before a meal. But then I went up to 2 capsules and boom, it hit me. My appetite was very low. I could barely get through my steak breakfast.
Yeah. So it's an interesting one. So what you're saying is not uncommon, and so we target the gastrointestinal tract, right? So what the way Amarasate works is it goes past the stomach into the small intestine and it releases. And that's where the cells are, the L cells that will release the GLP-1. It's why you need to have it, half an hour, 60 minutes, 90 minutes before a meal, because you want to get it through to that part of the gut. Now, the benefit of targeting the gut is that there's a safety profile, so we don't have to absorb it into the blood, so we don't have to worry about things like liver and kidney toxicity. You don't have to worry so much about drug interactions. So there's all these like sort of good safety profiles. The downside of targeting the gut is that the gut is much more variable between people than blood volume is. So you imagine if you have someone and you're absorbing a compound into the blood, people's blood volumes are roughly the same, right? So maybe a little bit higher or lower, but they're roughly the same. So it's quite easy to dose. With this, people's guts are quite very different. And so dosing is a lot harder. And so what we find is that the best way to approach it is to start low and to build it up, because we just don't know. There's no good marker of when someone's going to hit their sensitivity threshold. And that's a little bit to do with, one, how many basically taste receptors you have in your gut, but also, how good your stomach is at emptying, how long your small intestine is, or they're all very variable things. Yeah. So what we do is we start off with 1 a day for a couple of days, and we dose up to 2 a day, and then for some 2 at a time, and then for some people you may need to go to 3 at a time. Yeah. So it just depends a little bit. We get the odd person. So we're just running a clinical trial right now, 150 people, and we've found that and 2 people have been sensitive to the treatment, so that we've had to give them a special half dose. So we're looking at around, and they finally got up to a full capsule, but they're sort of a little bit more sensitive. So there's that sensitivity profile, and it's just a effect of how we're targeting the gut and that variation. Yeah.
Gotcha. So do people become less sensitive to Amarasate over time? Like, do you build up tolerance to it?
Right. So we don't see any evidence of that. What we know from, clinical research looking at how this mode of action works. So the targeting bitterness to the gut and stimulating GLP-1. So what we know from clinical research is, one, people don't become resistant to GLP-1 at a physiological level anyway. So you don't, if you're getting a stimulation then a reset, there's no indications that GLP-1 becomes less effective, right? So we're not worried about that side of things. If we talk about the bitter taste receptors, right, there's not a lot of work done in the gut, but what we have seen is that there is if you trigger the taste receptors in cells anyway, they upregulate them rather than downregulate them. So if anything, there would be an expectation that sensitivity may increase over time rather than decrease, right? But so, we're not likely to see a decrease. Yeah.
So if sensitivity increases over time, how do the people you mentioned work their way up to 1 capsule when they work their way down from 1 capsule to half a capsule?
Yeah. So I think there's 2 aspects to it. If we're talking about long-term adaptation, right? So upregulation of GLP-1, I guess, response, then that's a possibility. It's only shown in cells, so we're not talking about clinical trials here. We're just talking about a cell. If we're talking about the side effects that people get used to, the side effects tend to be a slightly different mode of action, right? So you'll get a few things like a direct delay in gastric emptying. So if you get some leakage of the Amarasate from the small intestine up to the stomach, then the stomach will shut off. It's bitterness in the stomach will cause it to not empty. So that can cause bloating effects. If you get the Amarasate slightly further down, you get a sensitization. You basically get an influx of water into the upper large intestine that can cause a flushing effect. And both of those appear to be, and they're not sort of a GLP-1 mediated effect. They're a secondary side effect, like from bitterness in different areas. And so those appear to, people get used to them over time. Whereas we're hopeful that GLP-1 effect is not diminished. Yeah.
Yeah. And is that also why I couldn't just break open one of the capsules in my mouth?
Well, you can break it if you want. I mean, so if you put it directly into the stomach, what you'll find is that it will be one, less effective. So you'll probably have to take more, and you're more likely to have side effects as well. So the particularly sort of a bloating, and what it will do is it will make your stomach pump a bunch of acid into it and try to get rid of that bitterness, and so you can get bloating, and yeah, so it can still work if you take enough of it.
That's if you take it normally. If you break it open and put it in your mouth, then it's going to do that. Or if you take it normally, and does it break down in your stomach? Where does it break down exactly?
No, it goes down to the small intestine. So what happens is occasionally people will get some reflux into the stomach, and that's when they tend to get that sort of bloating stomach effect, right? If it opens directly in the small intestine, then you shouldn't get that. If you were to break it open and swallow it, you would get a stomach effect, which tends to be that bloating. You'd also have a horrifically horrible taste in your mouth. It is quite terrible. So we get, this is quite an interesting story. So we get quite a prolonged GLP-1 response. So when we first did the work, we thought we'd get like a boost, like imagine this is the normal level, it will go up a bit and then it will go back down normally. What we get is we get a sort of a doubling of response, but then an extension as well. So it's lasting longer. And we think that's because the bitterness in the product, which is triggering the GLP-1 release in the gut, is very, very persistent. So if you break it open on your tongue, what you get is you get the slow buildup of bitterness, and then it just will not go away. And you can't wash it away. You can't get rid of it. It just sits there for hours. Yeah. And so I don't recommend you do that. I mean, you're welcome to. It's not going to cause you a problem, but you can't wash it away with water. You can't wash it away with alcohol. It's really challenging.
When you say bitter, I'm assuming you're talking about more than, say, a grapefruit, more than berberine powder. It's probably extreme.
Yeah, so we did, in the very first study, we did a test. We actually did an oral test before we enrolled people in the study, and because people have, I guess, what you call, have you ever heard of a super taster, like a bitter super taster? Yeah, so that's what that is there's a taste receptor, one of the bitter taste receptors. It's called TAS2R38, but there's a few different genetic differences between people, and it causes some people to taste it and some people not to. And so we were concerned, I guess, that our hop extract might be tasted by some people and not by others. So we got everyone enrolling in the study to make sure they could orally taste it, which meant if they could orally taste it, their gut should taste it too. And we got them to fill in a form that said, how bitter do you think this is? And it was from not bitter at all to the most bitter thing I've ever tasted. And pretty much everyone was right at the top there. One person came back and contacted me the next day, and he said, I can still taste it on my tongue.
Wow. Okay.
Yeah. So
I will not be breaking it open and putting it on my tongue then.
Yeah.
So the actual extract looks like, I want to say honey. It has like that color, and then it has that consistency. It's not like it's not a liquid. It's not a powder. And also, does the capsule have like a liposomal or some kind of coating on it? How does it pass through the stomach without breaking down there?
Right, so it's a modified cellulose capsule. So it's technically a delayed release capsule. So it's not a true enteric coated capsule. So you can get capsules that are resistant to stomach acid completely, but they tend to have some quite nasty chemicals in them. And so they're not really, you don't really want to use them. So we have a modified cellulose, which is delayed release, which is pretty good at releasing in the small intestine. It's not perfect, but it's pretty good. When you look at the raw extract, what you find is that it's quite waxy. So it's almost like a wax in its raw state. And so when we did before we did the clinical research, we did a series of what you call in vitro digestion models. So basically we got sort of shakers and I guess enzyme solutions that mimic different parts of the gut, and we put the extract into those to see what would happen. And what we found was that if you left it in its raw form as a wax, it would just sit there and it wouldn't disperse at all in the gut. So it has a formulation that's been done to it that just allows a little bit of, so that's why it's a little bit runny, but not really. It's still quite thick. So what we've designed it to do is to disperse in the gut a little bit, enough so it can interact with the outside of your gut lining, stimulate the release of those hormones, but progress through as quite a, I guess, what you call a bulbous mass. You can imagine it's going down the gut as a bulbous mass, triggering all of the hormones as it goes. If you were to leave it purely as that sort of wax, it just won't disperse and won't interact. And if you dilute it out too much, then that efficacy becomes reduced, I guess. So as it's going down the gut, it's getting more dilute and dilute anyway. If you start it off more dilute, then it dies off more. And certainly in the clinical trials, when we've given a stomach targeted one, which would cause it to be more dilute, and when it gets to the small intestine, the efficacy is not as long lasting. So it sort of dies off. So it's quite, I guess it's quite a careful formulation from that point of view.
Clearly a lot went into making sure that it's delivered perfectly. Speaking of, since it's so insanely bitter, will that interact with like what should be pristine environment of the small intestine? Like, will it impact with any of the microbes that may or may not be there?
Yeah. So, I mean, you shouldn't really have a lot of microbes in the small intestine. So what we know from the small intestine is probably pretty good. The more of the question becomes the large intestine, where you've got all your bacteria going. So we don't have a lot of information on that. What we do know from animal studies is that if you give hop compounds in the they've been done in rabbits. We didn't do the studies. I'm not a big fan of animal studies, but previous people have done them, and they've given these compounds to rabbits. What they've seen is that they are metabolized primarily in the colon, and none of them come through in the feces. So there's certainly no persistence in the gut. We don't see any overt effects on intestinal health as far as we can tell. What I will say is we are likely to be triggering GLP-2 release, or in fact we almost certainly are, and GLP-2 is a sort of a gut health hormone as well. So there's certainly some indications that we're boosting up some of the gut health hormones, and they would hopefully offset any negative effects you might see in your colon.
Nice. Yeah. There's not much talk about GLP-2 yet, but perhaps there will be in a handful of years, hopefully not decades.
Yeah, well, again, and I think that's one of the strengths of a product like this is that when you look at your enteroendocrine cells and causing this natural GLP-1 release, because those cells and those little same secretory vesicles also have GLP-2, you're secreting GLP-2. I mean, at some stage, I'm meant to be measuring some of our frozen blood samples to see it, but it's sort of one of those things where you're like, well, it's going to be there, so why am I spending my time measuring it? So we'll probably do that. But again, for the injectables, because you're suppressing, that one paper says you suppress GLP-1, but you also suppress GLP-2 as well, and so that's a negative on the gut health side for them, or potential negative where it's potentially a positive for us.
Would Amarasate have any impact on the brain?
Yeah, so I mean, again, if we're triggering GLP-1, then there's likely to be effects. You'll likely have possible effects on things like dopamine, serotonin levels, and indeed a possibility addictive behavior. There's also likely to be changes in vagus nerve activation and heart rate variability. And we're currently doing studies where we're looking at that. I think that ultimately in the future it would be really nice to do a big measurement on sort of addictive behavior with a product like this to say, can we do things like stop alcoholics from drinking or smoking, and we get a lot of anecdotal evidence about people reducing their urge to drink. And in this study we're currently running, so we've got a big weight loss study running, we've had that exact same comment from people who say, I was drinking 3 beers a day and suddenly I'm not. I just don't have the urge to drink the beer. So certainly in a subset of the population, we're observing, I guess what you'd call brain effects linked to addictive behavior.
Gotcha. And so if this has an impact on the vagus nerve, it seems like it would probably have a similar effect to the other GLP-1s, and it might increase resting heart rate and lower heart rate variability. But because this has a shorter half-life and it's not going to be 160 hours, is it feasible that if you took this in the morning, those metrics could start to return to normal by the time you're going to sleep?
Yeah. I mean, we'll get more information, but also this is a much more gentle approach. So, I mean, I think that you can we would be, well, I'm hopeful we're in a bit of a sweet spot where because we're doing a physiological approach rather than a pharmaceutical approach, that we're not really pushing a system into an area where it doesn't want to go and having too many of these effects. You're quite right, that's possible. We'll get more information from that in one of the current studies we're running, looking at heart rate variability and and again, we've got resting heart rate measures, so full spectrum of that with treatment.
Yeah, and since this is being used, it's consumed and goes through the gut and then into the small intestine. Are you seeing a lot of differences between different, like men and women, and then age ranges, and like what are the different populations that seem to respond differently, if there's any general trends?
Yeah, it's interesting. So we've done a couple of studies on directly comparing men and women for hunger rates and during fasting. So during a 24-hour fasting trial. And what we see is we see effects in both men and women. We also see, to be fair, we see probably stronger effects in women when it comes to suppression of appetite. And that's not entirely surprising. So what we see when it comes to appetite weight loss related measures, women are more sensitive to the GLP-1 injectables. Right. So they're no more. So if you look at the other benefits, things like, for the injectables anyway, for things like heart protection, insulin, glucose control, all of those, men and women are the same. But when it comes to weight loss and appetite control, women have a bigger effect due to GLP-1, and that looks likely to be linked to estrogen levels. Right? So there's actually a study in rats where they've given a GLP-1 injectable and they've conjugated it with estrogen. And if they conjugate it with estrogen, then it's a lot more effective than if they give estrogen by itself or GLP-1 by itself. So there's sort of a synergistic response. So that's likely the mode of action. Women are also more sensitive to bitterness than men. So we're likely, because we're targeting a bitterness response, we're likely seeing that elevated in women as well. So what we're seeing, I guess, is a fundamental likelihood that women have a slightly higher sensitivity to this product than men. What I'd also say is that if you're a guy and you're not feeling it, you can just dose it up a little bit more, right? So there's no absolute reason. It's not saying it won't work in a guy. It's just you may need to use a little bit more, and that's probably through two modes of action. Yeah. As far as things like with age go, we haven't seen any overt effects of decreased effect with age. Certainly, I mean, there are some studies that say that GLP-1 is reduced with age. There's also a study that shows that GLP-1 in the gut is increased with age. So it may just be not a issue of not having the GLP-1, but not being able to release the GLP-1 as you get older. If that's true, then something that's stimulating your endogenous GLP-1 is probably quite a useful tool in older people. So we're not seeing any issues with older people so far.
Okay, and then if you were to design a stack to amplify some of these benefits, whether it's satiety, it's to reduce cravings, are there any foods or supplements or lifestyle behaviors that you consider for that?
Goodness, a stack. Oh, interesting. I mean, so what I'd say is that I'm a big fan of, if someone's on a weight loss program, right? So you're using this to control your appetite and make healthier food choices. The biggest issue you get is muscle loss, right? Mm. Now, we're hopeful that we won't see as much muscle loss as some of the other treatments, because we're more physiological. There's other hormones being triggered, which may have some sparing effects. But I would always, if someone wants to stack a supplement with any weight loss program, I'd always recommend HMB. So I'm a big HMB fan. Yeah.
HMB over a complete protein source or even essential amino acids?
Yeah. So I mean, there's no issue with taking a complete protein source or essential amino acids, or even a branched chain amino acid supplement. They all do a similar thing. HMB is just more focused, right? So, and when you look at the data, I mean, HMB was used in hospitals to prevent muscle wastage during cancer and for burn victims, and so they used to use that before they started using things like growth hormone, which of course growth hormones are more effective. But if a supplement is good enough to be used in a hospital, it's usually a pretty good indication that it's got bioefficacy. And when you look at HMB, you look at it's a leucine metabolite. So you think of it, I mean, for the people who are listening, if you don't know, in your muscles, you've got 3 branched chain amino acids, leucine, isoleucine, valine. They make up the main structure. And then you sort of have glutamine and taurine floating around as well. When your body breaks down your muscle, when it breaks down leucine, it turns it into HMB, and HMB then signals basically, hey, you've broken down some muscle, don't break any more down, right? And so if you take that externally, then you can basically help prevent muscular breakdown during weight loss. The other thing I'd say is that there are some people have concerns around dosing up too much protein because it may be pro-aging. So there are things like mTOR activation. And I saw a study where they were looking at lifespan of rodents, and they were giving different amino acids, and they didn't show a decrease in lifespan when they gave leucine. Right. So obviously if you're giving leucine, breaks down into HMB, so that but they did see it with the sort of the other branch chain. So it seemed to me that this particular branch chain leucine derivatives were more safe to use, and really good targeted ones. So I'd recommend HMB for that reason. I take it all the time. I'm taking it now. What else to take with it? That's a good question.
What about something like the dietary basics? Like, I know that fiber might work on different mechanisms to help increase satiety. Perhaps maybe not supplementary fiber, but just a diet that contains fiber.
If you're looking at stimulating your gut so that your gut is more capable of releasing something like GLP-1, then any sort of green veggies, are going to be really good for that. They're going to increase it. If you want to take a probiotic, absolutely. Probiotics and prebiotics all have moderate effects on increasing GLP-1 levels, and particularly and possibly even, if you're looking at fiber, possibly even increasing the number of enteroendocrine cells, so the cells that release the GLP-1 that are present in your gut. So that's certainly something you could supplement and try with it as well. Yeah.
What about postbiotics like butyrate or propionate? Any of those?
Yeah, I mean they work in a completely different part of the gut, so they're really large intestinal rather than small intestinal. You can take them. I mean, very much if you were supplementing it, I'd want it to be targeted to the colon, and then they have they can have some appetite suppressing effects, and they can have some effects on the enteroendocrine cells as well. There was a study, must be close to 10 years ago, where they conjugated the different short chain fatty acids, so the butyrate, acetate, propionate, to a fiber, so that it would target it to the large intestine, to the colon, in rats, and they could show that they would suppress appetite and induce at least some weight loss with that sort of targeting. So as long as it's targeted to the large intestine, they're all good.
How about exogenous ketones? I know those don't directly impact the small intestine, but they do have an anti-appetite effect also.
Yeah, they're an interesting one. I haven't read a lot about exogenous ketones, but they certainly appear to be an interesting option for suppressing appetite. And really, again, going to a point where you're telling your body that you haven't eaten and there's no food around, so there's no point making you hungry anymore. And I guess that sort of lines up with that whole fasting thing, where people get an increase in hunger. Then if you fast for long enough, then that hunger goes away because there's simply there's no reason for producing it. But there'd be no issue with taking those. Absolutely.
Okay, so a little stack, Amarasate containing supplement with HMB, some exogenous ketones, and perhaps a little extra fiber in the diet as a simple way to reduce appetite.
Yeah, absolutely. Yeah, you'll be super slim in no time. Of course, you're winter there, so you've got plenty of time to get lean for summer, right?
Yeah, exactly. And then I know you spent most of your time researching what bitter substances impact GLP and the whole class of incretins, those hormones. Are there any things outside of bitter that have you mind blown or at least fascinated?
Yeah, so I mean, we looked at a few different approaches. So we looked at things like for triggering things like GLP-1. We looked at carbohydrate malabsorption. So you can trigger GLP-1 release by by getting, say, glucose into the very lower small intestine. And so we did some interesting studies on that where we wanted to say, well, how much glucose do you need to get into your small intestine in order to stimulate GLP-1? So we got some young guys, they're always university students who volunteer for these studies, and we put a tube down their nose, down their throat, into their small intestine, and then squirted some glucose in and measured their GLP-1 response. So you can trigger it with glucose. It's, and if you do carbohydrate malabsorption, so something like there's a pharmaceutical called acarbose or acarbose, which, is it's like sort of a alpha amylase, alpha glucosidase inhibitor. So it stops, glucose or starches and more complex sugars breaking down into the basic sugars that are absorbed in the gut. That can have an effect on GLP-1, and it would be really, really interesting to see if we can get something natural that's really effective enough to get some degree of malabsorption into the small intestine, to really lower small intestine, to trigger that GLP-1 with carbohydrates. We've failed thus far. We've tried a bit. It's not easy, but watch this space. It would be a nice synergy to get hold of if we could do it. Yeah.
And I'd love to see more safety information about, this doesn't seem like it's a mechanism that can naturally happen. I mean, it shouldn't naturally happen if someone's healthy. Is there a way that you could get a good amount of glucose down there that would be enough to stimulate GLP-1 release if someone's healthy?
Yeah, so I mean, what they'll say is the proponents of it will say, oh well, it's like if you had a really, really huge meal, that your body is, you've just eaten so much your body can't absorb all the glucose. I'm not a fan of that. I don't think that's true. The body has some pretty major glucose absorption capacity. It's really hard, and that's why we've struggled to get it to work, because we'll get a supplement that sort of blocks like 90% of uptake, and it's just, there's still so much excess capacity to absorb glucose in the gut or carbohydrates in the gut that you can't do it. So what we do know is that if you look at from studies where people have had parts of their intestine removed, and that you can or they've got you can really see that the likely safety issue is small intestinal bacterial overgrowth. So if you're delivering glucose down to really far down, then you can stimulate the colonic bacteria to grow back up into the gut, which is not a good thing. It's quite easy to measure in a clinical trial. You can do like a breath test where you measure changes in methane and carbon dioxide, and you can see whether you're getting small intestinal bacterial overgrowth, and you can also get what's called they call it dumping syndrome, which is a bit of a flushing effect as well. So there's, I guess, two quite good measures for safety that we would do if we were going forward in a clinical trial. But you're quite right; it's not likely to happen for a healthy individual. Yeah.
What about other entirely different ways of approaching this? Like, for example, you mentioned earlier that DPP4 is the enzyme that breaks down GLP, I believe. What if you were just to find something that would naturally inhibit that enzyme, and maybe that enzyme is also responsible for a lot more than just breaking down GLP? I'm not sure.
It breaks down a few different gut hormones. You can. So a lot of phytochemicals or plant compounds have DPP4 inhibition abilities. And certainly you could have a degree of activation or degree of ability to inhibit. The problem there is that if you target the gut, you can get a compound, as long as it's acid stable and gets through the stomach, you can get it into the gut and it's pretty much what you think it is. If you have to get it into the blood, then quite often compounds are changed. So the compound I like to think of is, I call it the phytochemical gold molecule. It's called quercetin, right? And quercetin is amazingly bioactive in the lab, and it is somewhat bioactive in people, right? And the reason for that is that quercetin is as it's absorbed into the blood, it gets changed and it gets conjugated, and it becomes quercetin glucoside or quercetin glucuronidate, quercetin sulfate. And the bioactivity is different. So if you're targeting DPP4 and you're trying to get your phytochemicals into the blood, chances are they will be changed. And so their DPP4 activity may or may not exist. You also need to have quite major inhibition as well. So what you tend to find is that these enzymes in the body are usually you'll need to inhibit them quite a lot before you have a functional effect. And so, you'd be asking a lot of a phytochemical to do it. In saying that, I mean, the Amarasate extract has DPP4 inhibitory effects as well, right? So we've measured it does inhibit it too. It doesn't, not a lot of it gets into the blood. So, how much we're actually getting there. But you're quite right. There are other approaches. They work pharmaceutically. Watch the space. They may work with a phytochemical nutraceutical in the future.
Ed, what's in your stack, whether it's for GLP or just in general?
Oh, right. So what do I take? So right now I take a protein supplement, but I take an HMB. I'm taking a gram of resveratrol, 500 milligrams of quercetin. And I take the quercetin not to get into the blood, but try to help it get the resveratrol into the blood, because resveratrol again is sulfated as it's absorbed. And so if you take something like a quercetin, or you could take something like a biopiperin or something to try force it into the blood unmodified.
Do you like resveratrol over something similar like pterostilbene?
Yeah, I mean, yeah, they're both good. They both probably work the same way. I like resveratrol because I can get lots of it for inexpensive. But they all have similar effects. I also take some spermidine supplementation. I did a really interesting one recently where I took a 16 chain long fatty acid. Yeah, and it was sort of it's meant to have all sorts of it made me bleed lots. Actually, it was really weird. Apart from that, that's all I'm on at the moment.
Is that 16 chain fatty acid? It's not C15. C15 is 15 chains.
Yeah, C15's 15 chains. Yeah, it was. Trying to think what the name of the supplement was. But anyway, it was one of these science conferences. It was just being sold, and I just gave it a go. It was really expensive. So there's just those ones. I mean, in the past, I've taken a huge amount of supplements, but I'm really good with my diet. So I'm not supplementing like vitamins and things like that. So I'll eat. My diet consists of green veggies, some lean meat, black or purple rice. So lots of anthocyanins, kiwi fruit every day, berries. So it's a lot of, I guess, what I'd call healthy foods and limited supplementation. And when I do supplement, I tend to take huge dosages. The other thing that I've taken in the past, which I'll use intermittently, is obviously creatine. But there's a supplement, the ectosterones. So I'm not sure if you've heard of those before. They're an estrogen beta agonist. And so they basically, there's some good studies showing they stimulate muscle growth. But they're hard to get pure. So what you find is that a lot of the time you'll buy like 100 milligram dose, and it turns out it's only got 1 milligram in it. And so I did a really interesting thing where I read a paper where they showed if you took 200 milligrams or 600 milligrams of 20-hydroxyectosterone a day, you could generate like pretty decent muscular growth. So I went and bought some. I got some pure, through sort of scientific links, and I thought, oh well, they're taking like 600. I'll take a gram a day because got to up it a little bit, and I got terrible side effects. Yeah, and so what happened was that in the study, it turned out that the supplement they'd used was only 10% pure. So it was only 10% what they said it was. So I wasn't just taking more; I was taking like way, way more. And even though it was a estrogen beta agonist, which is muscular growth and things, it still has some estrogen receptor alpha antagonism, which is the feminizing one. And I was getting like itchy nipples, and they were all sore, and I was like, "What's going on?" And yeah, so that works really well, but just careful on the dosage.
That's a good reminder in general that when you see something in research, not just to take the dosage, and if it's in animal models, extrapolate it to humans, but even if it's in humans, that you have to be careful because their product they used as the reference in the study might not have been the same as the product that you're getting.
Yeah, exactly. It might not be the same, and there's no guarantee that just because they used it that their numbers are accurate, and that's really the big thing. Because I mean, one of the things that we do in plant and food research, which is whenever we're doing health research, we're required to do a chemical analysis of the product we're using to make sure we know exactly what it is. And sometimes we'll publish it with the paper, or refer back to it. And most studies won't do that. So they're just taking the supplier's word for it, or it could be a commercial product, the commercial product person's word for it that there's actually it is what it is. Yeah.
Wow. Well, Ed, if people want to follow your work, to connect with you, is there anywhere that you'd like to point them?
Yeah, so if you go to Plant and Food Research, so plantandfood.co.nz, and you search for either myself or for Amarasate, there's links to the research we've been doing, and there's updates. And if you're interested in the commercial product that's been made from my research, which I get no money from the sales from because I'm a government employee, and no money from the patent that was made either. That's the product is called Calocurb, and they have lots of social media type stuff you can read about there.
Yeah, and I've used Calocurb. 2 capsules works for me. I am an affiliate of theirs, so if you buy through my link and use the code URBAN, I do get a tiny commission. So I'll put that in the show notes for this episode as well. But Ed, this has been a blast. Thank you so much for joining me on the podcast today.
Oh, it's been a pleasure. Yeah, and you have some great knowledge. It's amazing, man. Good work. Yeah.
I hope that this has been helpful for you. If you enjoyed it, subscribe and hit the thumbs up. I love knowing who's in the 1% committed to reaching their full potential. Comment 1% below so that I know who you are. For all the resources and links, meet me on my website at mindbodypeak.com. I appreciate you and look forward to connecting with you. As a reminder, the information depicted in this video is for information purposes only. Please consult your primary healthcare professional before making any lifestyle changes.